So I should start actually by saying this is actually a collaborative project. Obviously, we're looking at hematopoiesis throughout development, and no one person can do it all. So it's basically been a collaborative project between my lab, so the Irene Roberts lab, Mead lab at Oxford, Dave Bodine's lab at NIH, and the Cara Dimitris lab at Imperial College London. And what we've done is, I mean, we know hematopoiesis is going on throughout the human lifetime, and it begins in the fetal liver by 5 weeks of gestation and carries on through life. But the fetal liver hematopoiesis ceases at birth, and fetal bone marrow hematopoiesis, although it starts at 12 weeks of gestation, doesn't take over as the main site till the baby is born. And so we wanted to kind of understand how the architecture of human stem and progenitor cells are changing through ontogeny, because surely there must be different requirements at different stages of life. And I think you can truly dissect this only when you look at a single cell level, because if you look at a bulk population, it's pretty much like looking at a handful of colored sand, but you don't know that handful of sand is made up of single grains of different colored sand. So we want to split those apart and see what proportions each different color particles make and in what architecture they're kind of arranged. So we started looking from very early fetal life. So we looked at fetal liver around 7 weeks gestation, and matched fetal liver and fetal bone marrow from later gestation, and then postnatal pediatric bone marrow and adult bone marrow. So at the moment we've used chromium 10x to look at the architecture of the whole stem and progenitor cell compartment, basically, and we can clearly see that the composition is different at different stages through life. So in early fetal life, for example, there's a large expanded erythroid compartment, which is what you would expect, because that's what the fetus needs, mainly red cells at that stage in life. And then postnatally, you have the other progenitor subsets, and the lymphoid subset is a bit more prominent in the pediatric marrow, which is, again, I guess that's when children develop their immune repertoire, so you'd expect the lymphoid compartment to be active then. But rather interestingly, we found when we looked at matched fetal liver and fetal bone marrow from the same fetus, so exactly the same developmental stage and the same human being, so to speak, they were markedly different. So the fetal bone marrow had a much more expanded lymphoid population and a myeloid population than the same fetal liver from the same developmental stage, which suggests obviously the microenvironment plays a big role in deciding how the stem cells should differentiate or which lineage they should go down. So at the moment we're exploring those, like what is driving those changes between specific stages, but also sites. I think we'll have to characterize this a lot further, because this is preliminary data giving us a broad overview of how hematopoiesis is changing throughout the lifetime. But already we can see several possibilities, because there are postnatal diseases that originate in fetal life. I mentioned infant leukemia, but also other diseases like juvenile myelomonocytic leukemia, JMML, and we think we might be able to identify the unique target cells for diseases like that, looking at early fetal liver, for example. The other implication, I think, is in postnatal life, we use mobilized stem cells, mobilized peripheral blood or bone marrow from donors for transplants. And I think it's really important we understand what we are dealing with. We take CD34s, we put it into another person, but inherently it may be different depending on what stage of life you're harvesting those cells. So we found, for example, mobilized peripheral blood to be quite different from bone marrow stem and progenitor cells, for example. And at the moment, like I mentioned, there are about 4 or 5 groups that are looking at it. But in parallel, we are comparing it with disease states at the same same developmental stage. So for example, I'm interested in looking at how fetal lymphoid development is different in Down syndrome, for example. So I compare normal fetal lymphopoiesis with Down's fetal lymphopoiesis. But my other collaborators are, for example, looking at JMML, like I mentioned, or DBA, Diamond-Blackfan anemia, or much later in life, MDS or myeloproliferative neoplasms, and kind of matching those pathological hematopoiesis to the normal data set we've got. No, I just wanted to actually say that I was lucky enough to get a TRTH fellowship through EHA and ASH, and I think it's a great opportunity for researchers, clinicians like myself, clinician scientists like myself, or pure scientists just about to set out on their independent career pathway. So I think it's a great fellowship.